side of the waypoint too, just the middle of the high.
Okay. So not to the right or
I
southeast on it?
Yeah, my guess is that's
Okay.
just a plotting error.
Copy that.
That's 100-meter.
Contours?
Right,
Gridded.
Oh, gridded, yeah.
Yeah, so it's not great bathymetry.
Yeah, copy. Okay. So what do you guys want to call it then? You're facing way off, so.
Well, we're heading south, right?
Yeah, more southwest than anything. So I would
You gotta turn this.
Do you want to turn?
get that guy around.
You push ahead, I'll turn.
So for those of you who are following along with us today, we are here at Pigafetta, Gio,
Yeah, it is.
and we are just moving along a small ridge. We think that this ridge may have actually slid or faulted off of a higher plateau. So it's sort of a small local high among a large flat area. And we've seen a really nice diversity of corals and sponges today. We've seen a few other interesting things like fish and urchins.
Do a 360.
And we have a lively ongoing debate about what the geology of this place actually is.
Copy that.
So continue to stick with us. You'll have a good time. It's about 2,000 meters down, 2,035 right now.
There's a huge one of these.
Whoa.
Yeah, so can we stop and take a little look at it?
That is
get
big.
Georgia.
Get the lasers on, please. Yes, it is. Look at you out there in front.
Lasers on.
What is it?
Iridogorgia is the
Nailed it.
genus.
Is that the one that Chris was saying could get over 5 meters high? Or is that something
it might
different?
have not have been in here, but maybe,
That's
yes.
the one.
Yeah.
Oh, sweet.
So how big is this one, pilots?
It's pretty big. The lasers are on it right now, so
I can't even
Oh,
see
yeah,
the lasers.
okay,
Where are
yeah,
they?
yeah.
I mean, just the part
This
with
is
polyps
just a
is
baby.
probably a meter.
Wow.
So this one's probably about 2 meters tall, 2.5.
Yeah, seems about right.
I mean, that's a pretty big coral.
But it's only as half as big as it could be.
Yeah.
Teenager or young adult.
Great, thank you, pilots. We can
Navigator,
move
you
on when you're
thinking
ready.
220, 210 in there?
Yeah. You all set for that, co-pilot?
Yeah.
Okay. Pilot has 210 sound.
Sounds good to me.
I'll line up on it.
I'm going to give you guys 40 meters. Bridge, nav.
Where am I? Am I off to your starboard? I have to push port.
Yep. Hey, ship move, please. Range 40 meters, bearing 210 degrees, speed 0.2 knots.
I'll bring you in view.
So I gotta go.
Good
I
copy,
have
thank
to
you.
Go around this massive coral.
Copy.
I'm coming over.
Alright. I'll turn back 210.
Let's do a pirouette around this. Just do a partial.
Coming partial, holding there.
That's good. We've seen a bunch of these today, I think.
In addition to the lovely sponge, sure looks like we're seeing lots and lots of disaggregated plates, probably of manganese oxide crust.
All right, coming full wide. Full wide.
Thank you. Come on in, video.
What is that, Diva?
Is that hold fast?
You're off.
So that just looks like the base of a sponge where it used to be attached, and it's obviously since broken off. I.e., move along. Oh, but there's a little squat lobster just shooting above it.
See what happens when you say move along? Squat lobsters come out of
Squat
nowhere.
lobster.
Or are you going to sing for us now?
Nope.
All right, come on up.
Coming wide. All right, full wide. 210. Line up on it. Just a couple more degrees here.
Let's do a partial here, video.
Partial.
Yeah, we can come in tighter.
Coming in.
Yeah, that's good.
Holding.
So again, we're seeing another dead sponge colony here, which is just absolutely laden with loads of other animals that have colonized this. We can see a couple types of corals.
Is that a crinoid at the top?
A couple types of corals, some hydroids, some camatulid crinoids. There's actually even a squat lobster living in the chrysogorgid coral, which is the pink one behind.
It is really amazing how they're utilizing these stocks.
Yeah. And it's interesting because it's not like there's not enough hard substrate here. It's just that these animals want to try and get up as far off the seafloor as they possibly can. So rather than live on, you know, some of the ferromanganese crust, why live there when you can live a little bit higher up the skyscraper
Yeah,
and
they're starting to move there.
in the penthouse?
Yeah, come on up.
Full wide.
So we have about 2.5 hours, 2 hours and 20 minutes left in this dive. Nav, could we have an update on how much distance is left to get to the end point and whether we're on track for that, please?
Sure. Stand by one. Let me calculate that.
Come on in, video. This might be just hold fast. Yep,
Partial.
hold that, yeah. This guy's not doing too well. Alright, come on out.
Full wide.
Okay, watch these. I have a little over 2 hours left, 2 hours, 15 minutes left. And that,
Come on in, video.
if we travel at 0.2 knots all the way there, no stops of the ship,
Hold that.
it'll take an hour. So fully expect us to reach waypoint 2 by the end.
Sorry, so can you repeat that? I got sidetracked by the alarm that's now going off on the ship.
Hey, come
So
on.
you said if we travel at, oh my goodness. Just, this is a drill, everybody listening on shore. It's absolutely fine. We are not abandoning ship. Anyway, so, or fire, or whatever it is. So,
Hey, come on in, video.
So yes, barring any major stops, we will make it there.
right, okay. But you said we need to move at 0.2 knots.
Affirmative, yep.
Come in tight.
And is that faster than we usually go?
No.
Okay, just
All I
checking.
got.
So, Chris, I'm not sure if you heard that, but we are well on track and should make it to our final waypoint.
We have a little over 2 hours on bottom, and it should take about an hour to get there.
Thanks, Nav.
Sure.
All right. Come on out.
Full wide.
Quick zoom here. Hold that. Dead sponges, maybe? I don't know what that is.
Yeah, I think this is a great time for us to do some little, history lessons on where the name Pigafetta's come from.
Hello, everyone. This is Lindsay McKenna.
Quick
I am
zoom
the
here.
mapping lead on
Come in
board,
tighter.
and most of my operations are done overnight. So yesterday, when we were making a plan to map our way over to the Pigafetta Gio, I was a little interested in where the name came from. So I did a little research, and this Gio is
Okay,
named
come
after
out.
Antonio Pigafetta, who was an Italian,
Full wide.
or as Steve would like me to say it, Antonio Pigafetta. We did a little pronunciation practice on the ship yesterday. But Pigafetta sailed on Magellan's first circumnavigation voyage around the world. And on that trip, he collected extensive data on the geography, climate, flora, fauna, and inhabitants of the places that the expedition visited. So he was a real explorer,
video.
and it said that his meticulous notes were invaluable to future explorers and cartographers, which is what we are. So I made some very early maps of not only land, but the seafloor and coastlines, and collected a lot of
Come
information
in tight.
about the biology of these new places.
Great. Thanks, Lindsay. So, you may not know the answer to this question, but maybe you do.
Got a little bit more.
Do we know who named
Yeah,
the
hold
seamount
that.
Yep.
after Antonio Pigafetta?
I don't know the answer to that, but I do know that tomorrow's dive is named Enrique, and the one after that is Del Cano. Oh, okay, I won't tell who they are.
You're going to have to save that for each dive. You're going to have to come in here tomorrow and the day after. So we were just looking at a ophiuroid sitting on the seafloor. It looked like an ophiocanthid ophiuroid, and these are usually commensals on these large sponges or corals, so perhaps it was just,
He jumped.
he jumped off of... Did you actually see him jump?
Yeah, I saw him land.
Yeah. So I don't know why he just jumped ship, but he did. Yeah, jump ship. And there's also this beautiful comatulid crinoid sitting on this, what looks to be quite dead isidid skeleton,
I'm at 80 degrees. You got a little bit of time.
which also has some hydroids on it.
Hey, come on out,
Anyway,
video.
so thank you, Lindsay. So we'll see you same time tomorrow.
Absolutely.
Full wide.
And in addition, it looks like the name was proposed in 1985.
Yeah, going uphill again.
Yeah. a pretty big one.
So that crinoid was likely Antidonidae or Thalassometridae. And here we have another one of these really large corals. Nav, what's it called?
That's a Rudogorgia, Washley.
Exactly. I don't even know why I'm in this van. Iridogorgia is the genus.
And the ROV pilots know it better than we know it.
Negative bridge.
That's beautiful.
And these are these very characteristic spiraling whip corals. Oh, everybody's sneezing in the back here.
Very nice pilot.
So I assume our goal is to keep making way towards the top of the ridge.
Yeah, so we just got an update from Nav a little while ago, and we're making great time. We have 2 hours left on bottom, but this should only take us an hour to get to our final waypoint. So we're just going to keep going, observing the brilliant biology and also great geology.
All right, come on in, video.
Awesome.
Oh,
did I not
okay,
see
so
this
this,
Almost went right
It's a
by
fish.
us.
Bruce, are you watching? This looks like one of those gnarly fish which we were seeing before, which you didn't know what it was. And that one looks a little preggers too. Pregnant.
Come on in, video.
Or it just had a very big meal. Okay, no. So Bruce is saying this one is likely spectrunculus. So it's a type of
Oh.
cusk eel.
Did it just run into the rock?
Yeah, I mean, they can't see very well. Especially when you shine
Bright lights
really,
at them?
really, really, really bright lights when they have no light usually into their little eyes, which you probably don't work that well either. So yeah.
It looks a lot like you getting around the ship.
So, isn't that funny? So, likely spectrunculus. Thank you, Bruce.
Losing the light.
Yeah, I'm letting him go. It's going off to my port too much.
Come
Come on out.
We ready for another ship move or?
Yeah.
Or what?
What do you think about that heading? You want to change a little to starboard, make it a little...
Well, we're going uphill right now, but I'm down with anything. Whatever you guys think.
What do you think? Co-pilot, maybe 230? How's that sound?
Yeah, let's do that.
Okay.
So it's a little surprising to me that we saw such a pretty high density of corals and sponges and other things when we first landed. And as we've been moving up this ridge,
Come on in,
it's sort
video.
of kind of come and gone. Chris, do you have an idea maybe why that is or
Bridge,
what
yeah.
we might expect to see?
Hold that.
What? Is that thing floating? What is it doing?
Hey, can we have a
It's
ship move,
holding
please?
on by very, very little.
Range
and you can see it's
40
also
meters.
regenerating.
Oh,
Bearing
yeah.
one of its legs.
230
It's the
degrees.
tiny leg.
Speed 0.2 knots.
And it didn't lose the whole leg, it just lost a piece of the leg, which is now regrowing.
Oh no!
Oh, no!
0.2. Thank you.
Oh, hard landing! Hard landing! Oh.
It probably took him like 3 hours to get to the top of that. More. And now he has to
Oh,
start
this is so
all
sad.
over again.
It's devastating.
He's just lying there, totally defeated.
Ship move in.
It's like a little person.
I think he's checking out all his limbs to make sure he's okay. Wiggling one, then the other. But seriously, folks at home, we do not try to anthropomorphize our
Yes.
organisms here, except when we do, which just happened.
That's the third one we've seen.
Jump off.
Bust a move.
Yeah.
Yeah,
And this is the only dive we've ever seen that on, right?
Yeah, I think so.
We typically see that. We just haven't seen it much this expedition.
Yeah, during this expedition,
Yeah.
we haven't seen it before.
They make parachute drops when they
They make
feel
what?
us. Parachute drops
Oh.
off their perches when they feel us coming.
Well, I have to say the event log was just as giddy as we were by watching this guy fall off. There are actually some somewhat funny comments. Good job, guys.
do a quick zoom here, video. Another sponge that fell.
So yeah, lots of these large dead sponges, but this one is actually coated in manganese crust as well, it looks like. So this may have been here for quite some time, because as we were discussing on one of our earlier dives, you know, this ferromanganese crust forms at a very, very, very slow rate, typically 1 to 5 millimeters thickness per million years. And you can see growing on it, it's got a chrysogorgid, a comatulid crinoid, a couple other things.
Come in tight.
Ooh, are these? So Chris, do you know what these are? Are these stolonifera corals?
There you go.
On the underside of the sponge? Good spot, pilots.
Thank you.
How interesting. Okay, I think all of our comms are down, but that's interesting, and perhaps they're stolonifera corals.
Shore-based science team, can any of you hear us? If you can you respond to us or put it in the event log?
Yeah, I'm gonna feel pretty happy about my guess of stolonifera.
That's it.
And this is when someone's gonna chime in and be like, "No, David, it's a zoanthid."
Stoloniferian?
Yes. Did someone write that?
Yeah.
Yay!
Okay, so it sounds like the feed is actually coming through, Diva, so people
Oh,
are able
can we
to
zoom
hear.
on the purple polychaete behind? Is that the best zoom we have?
It is.
You want to zoom on its behind?
I can get closer.
Maybe.
Come on out.
So that was a beautiful little
Get you a closer shot.
scale worm polychaete, polynoid. And their colors always amaze me.
Alright, come on in.
Holding.
Right. Do you have any more?
Full zoom.
Beautiful. And I was saying a couple of dives ago that it's always great to see these intact on the sea floor because they have these scales on their dorsal surface, which when collected, they usually drop. And so you hardly ever actually bring them up to the surface if you sample them with their scales on still.
How are we doing?
And their scales are one of the identifying
starting to move here,
features
so-
of these polynoids.
You're still pretty far in front of
It's
me.
almost iridescent teal.
27
So
degrees.
beautiful.
That doesn't sound like a defense mechanism. Is that a stress reaction?
30 degrees.
I am not sure, perhaps.
Alright, come on up.
Polynoid.
Full wide.
Get this sponge as I go by.
Okay.
Let's do a partial.
Holding.
Alright, come on in.
Holding.
So we're seeing that coral behind the sponge, and then is that another crinoid on the bottom? What is that?
Yes, another crinoid living commensally on the sponge stalk.
Sorry. Alright, come on out.
You'll be happy to know we all survived our
this
fire
is
drill.
a different species of Caulophacus that we have collected before in Hawaii, but this is the first time we've documented the species here.
Great, thanks, Chris. So does that species have a name, or is it just sort of a morphotype or number?
Coming up.
Coming up.
Or not.
Should be at the end of that ship move, guys. You want to get another one? Up?
It
Oh, look,
doesn't
lots of corals.
have a species name, and I'm just checking right now, and I don't think it even has a subgenus name yet.
Bridge now. Okay, copy that. Let's put another one in if you're all set. Range 40 meters, bearing 230 degrees, speed 0.2.
Come in partial video.
Good copy, thank you.
Hold that. Yeah,
I'll take you off the XRV next time, sorry.
that's okay. All right, come on out.
So again, it's just most of what we've been seeing on this dive are quite large, bushy, isolated corals. We've also seen a couple iridogorgid corals, whip corals, as well as some pronoids. And we've also been seeing a good deal of quite large sponges. And this is definitely what we were expecting to see on this dive. We do understand that the video feed has not been great during this dive, and we appreciate all of you that have stayed with us, onshore scientists and also the public.
So the few of us geologists that are on this dive have been spending a lot of time speculating over whether we're looking at old, heavily sedimented manganese-crusted pillows or whether we're looking at something totally different.
Come in partial here.
So it's been highly enjoyable for geology on this
Yeah,
dive,
love
almost as much as the biology, which has just been fantastic.
In fact, Deb, I was wondering if somewhere around this area, if the pilots are able to set up, if we could try to go for a rock here.
Let me find out. Now this is watch lead.
Go ahead.
How fast are we moving right now? Like, could we stop if we wanted to take a sample or not?
We're moving point 2.
What kind of sample are you looking for?
We wouldn't mind taking a rock sample, but only if
Come
it
on
is
up, video.
exceptionally easy to get. You know what I mean?
You don't want to compromise time.
Right, exactly.
Sure, understand.
Do you want me to stop it?
So,
No, let me see if I can find
This
something that
really
looks
looks
loose.
like
Can you
pillows
ramp up
to
my
me, and
hydraulics?
is there any of the loose ones that are not cemented to the seafloor by manganese?
I don't know. There's a lot of cementation here, it looks like. Also, you know, I am a little cognizant of the fact that we definitely want to make waypoint 2. So I'm a little bit concerned about us stopping to sample, just because it sometimes takes a little bit longer than we'd like. So it would be good if we could get maybe both your and Chris's opinions on that, Patty.
Okay, yeah, I was wondering about I presume that at the top of this hill is likely
Take
also
down
to
my
have
gauge.
exposure similar to what we're seeing now.
Okay.
If you want to go for the end, that's fine.
I think that would probably be a more prudent path for this one.
It's a
Yeah, Chris is
huge
nodding
coral.
his head. Yeah, go for it.
It's a big one.
So to keep going for waypoint 2?
That is, go for the top.
Yeah. Okay.
Yes.
All right. And biologists, please shout out if you need us to zoom on anything. Or geologists. Anyone?
Yeah, we should make the time with some. Oh, should make the top with some time to spare. So if we want to take
Great.
one up there, then
Yeah, that's probably the preferred.
be a little better. I
Yeah,
think.
exactly.
If I get out ahead and I see something, I'll stop and grab it quickly, though.
Very cool. Thank you.
Yep. We've moved into the land of giants.
Yep.
I think the toughest thing is just going to be finding some place to sit down without hitting a coral.
Yeah. That's tricky.
There's
Rather.
a lot of big guys, a lot of trees.
Some of those might be loose, but I don't know. Could always nudge it first.
Yeah, I'm going to nudge a few of these. I don't know if they lose. It's really old seamount, so. I'd be surprised if anything's loose.
I don't see any movement. Yeah.
Definitely not loose.
Nope.
Yeah, nothing moved.
I'm looking down at you at 60
Yeah, pushing ahead.
degrees.
Watch the,
Yeah, I've been
Going
keeping an
up.
eye on it.
yeah.
I can see over it, so.
Okay, we just passed a rock in the bottom of the tree. Just making a note that it looks hollow and has the sort of fracturing that you typically see radiating out from the center of a pillow. So I think these things really are pillows. Certainly they're covered with magnesia.
Coming up.
Great.
Pillows.
I just tried to tap a few rocks, and it's looking like mostly cemented stuff.
Yeah, I would believe
I didn't
that.
see anything move.
Thank you. So, so we had our fantastic ROV pilot try to tap some of the rocks, and none of them are moving, and that makes a lot of sense because it does look like this is fairly cemented or indurated right here. So we'll keep moving on. We'll keep imaging these corals and sponges, and then hopefully once we get sort of very close to our last waypoint, which is on a local high on this ridge, we might be able to grab a rock.
You got closer
So again,
to
just another couple
210
more of these
navigator.
Yeah.
extremely
Copy that.
sort of bushy
How's that
isolated
look for you?
corals.
Local high still seems to be 250
Perhaps
for
Jason
now.
Isis was what was suggested.
Either way, I think.
Yeah. Maybe I'll just.
And then
Make
quite a
it
couple
220.
Iridogorgias
Yeah.
right here, all in one place. This Iridogorgia has a 1 or 2 Ophiocanthidae ophiuroids at the top of it,
Come on in, video.
trying to get just as high up as they can possibly get.
Pilot, I'm aimed on
And these are
220.
Ridogorgias here. We're seeing
Flames are
these
moving
these
there and
spiral whips are actually a type of chirostylid. Not what am I talking about? Chirostylid? That's a squat lobster. Chrysogorgid. Sorry. Disregard. Let's try that again. So
That
these
was awesome.
These beautiful spiral whip corals, known as Iridogorgia, are actually a type of Chrysogorgid.
Let's do a quick zoom here.
And what exactly is a chrysogorgid?
It's a type of octocoral.
Hold on,
Does it
partial.
mean something?
Yeah, come in
I
tight.
mean, there's going to be something to do with its morphology in
Hold
terms
that.
of certain characteristics that it has, but I am not a coral expert, so perhaps Chris or somebody else can explain. But
Perfect.
Yeah.
Great.
I can see one on the screen and be like, that's a Chrysogorgid, but I can't really tell you why.
Good.
And then,
Okay, looks like ship's coming
Yeah,
to a stop. I'm
come on
going to
out.
call another one in.
here we're looking at a stalk sponge. It looks like it's from the genus perhaps of Caulophacus.
Forward.
I'm not seeing the fibrous stem that denotes, for instance, the hyalinemids. But there's just, I mean, there are lots of very large corals and very large sponges, but there's just not much else, for instance, in terms of
A lot of twos.
invertebrates living on the deep sea floor.
Copy that.
It just seems to be these quite large sponges and corals and their commensals.
Well, if we are correct in thinking that these might be pilobasalts, which we're not wedded to at all, it would mean that there's only a fairly, light layer of sediment, and then you're getting back down into hard rock. But that would mean it would be good for them, wouldn't it? Not for the infauna,
Do
though.
a quick zoom on this.
It would be
But
good.
we
It's
wouldn't
good
see
for the
the
coral.
fauna anyway.
Right. It's good for the corals and the sponges and things.
Oh, okay. If you have any more, just tight.
So here we've got another beautiful octocoral with several Ophiocanthid ophiuroids living commensally on it. It almost looks like some of their polyps are missing on their branches and sub-branches.
Don't have too much time, pilot.
No.
For me to be.
We're
Could
happy
we
to move
zoom
when
out
you're...
on this colony just for a second when you get a chance? I just want to see the whole colony.
Come in partial just for the colony.
Got it. Okay, thanks. Actually, that's a coral we haven't documented so far today. It's a primnoid, and I think it's a coral we haven't documented so far today. It's a primnoid, and I think it's Calyptrophora angularis, so that would be a first observation of that, perhaps on this cruise.
So great, thanks. So this is a primnoid, as Chris just said.
Are there two different kinds of
Calyptrophora
polyps on this?
angulata, I think he said. And Scott's pointed out that there are two types of polyps growing on this coral, and that's because the coral itself is in quite bad shape. We were noting that some of the
About that
polyps are
time,
actually
guys.
missing,
I
and
gotta pull up.
the larger polyps off to the left are actually zoanthids, which are overgrowing what is likely a not very well coral. Calyptrophora angularis. Thank you, Chris.
Well, as we've been talking about, these really do appear to have a morphology that looks like pillow basalt. We don't really know that that's what it is. The rounding is also characteristic of what happens with a thick manganese crust, but it could be.
Yeah, you got a rock in your starboard and biospecimen and port outer.
Thank you, Nev. Okay, watch lead, new pilot here with you.
Cool.
I hear we're heading along the ridge. We have one bio and one geo sample.
Yep.
So I'll just continue along, and I'll keep my eye out for loose rocks.
Great.
Let me know what you see.
Excellent. And the loose rocks is less of a priority than achieving our waypoint.
Okay.
Thank you.
Welcome, co-pilot.
Pilot.
I haven't even seen.
Okay, so Nav, can you clarify the direction of the last move, or the direction that we're currently moving?
Yeah, direction last move was 220.
220.
So we're pretty much just following local high,
Okay.
which is right now about 220.
Yep.
Nice. And it looks like it's curving a little to starboard now, if you look at Sirius sonar.
Yeah. And how much left is in that move?
It looks like it completed about 5 minutes ago.
Oh, okay.
So you should just
Swing for
have a
few
little
minutes.
swing.
It's
Yeah.
like the ship's still going, isn't it? Yeah, the ship's...
Oh, sorry, it did not complete. The word has the word complete, but then colon with the
Oh.
time, but the time's left. I just
Gotcha.
noticed that. All right, so it is still in motion.
Okay. How long was it?
40 meters, so it's about about 10 meters left.
Okay, so we're still cruising. I'll keep out ahead of you, co-pilot.
Okay.
A lot of giant coral here.
So this is a particularly dense area of corals, and we can also see some of these really interesting sponges. I think they're farreids, but I can double check that.
This is beautiful.
So that skeleton-looking sponge, that's definitely a farreid, and it could be in one of two genera that I'm a bit confused about these days. Could be in the genus Aspidoscopula, or it could be in the genus Ferrea.
Sure, coming in.
Great, thank you, Chris. Just the sponge morphologies you get
Holding.
down
Yeah.
here and in the Hawaiian Islands on these seamounts
Creep
is
up
just
and in.
absolutely incredible. Again.
Okay, you can go a little closer.
Coming in.
And
Holding.
after you get your 10 seconds, I'll move on.
All right. You want to move on?
All right.
All right,
Go
coming
ahead.
wide. Oh, going on.
Oh, yeah, no, you can go.
Okay.
Yeah.
That's all she got.
All right. So we'll give 10 seconds here.
Yeah, that slope is really starting to move starboard. Here
Yeah, so the ship just finished.
It is done now?
Yeah.
Okay. All right.
Okay, whenever you're happy, video.
So coming if you wide. want to center up after this, we can adjust
Yeah.
bearing and put in another ship move. Full wide.
Thank you, video.
We're not too deep, so not too much of a swing. Just a couple minutes.
How far from waypoint 2 are we now?
So roughly a little over 200 meters, so not that far.
Okay. So
Get there in a few moves.
yeah.
What does HIPAC say as a bearing to waypoint 2? Just curious.
Exactly 200, but the general center area is what we're going for. Waypoint 2 is not a
Okay, so
hard
it's the
target.
center area.
210, so between, 200, 210, 220. But we'll probably follow local high to get
Yeah, just follow this ridge. So like
area.
240 looks to be.
Yeah, that looks, if that's where we are right now. Yeah, that's
Sure.
not too far off. That'll still get us in the right direction too, so it'll get us closer.
Yep.
All right, so I'll do 40 meters at
So as we take a
240,
stroll through these really beautiful corals,
Yeah. 40 meters,
I thought
240.
it might be
Copy that.
good to ask a couple of questions of Diva and Chris about the manganese crusts. And so I think we're still in the prime crust zone,
Bridge, this is Nav.
maybe right at the far edge of it. But what's
Can
so
I get a
special
ship
about
of,
these
40
crusts, and why do we
meters
care?
At 240 degrees at 0.2 knots.
Chris, do you want to go first?
Go for it, Diva.
Oh, I was just deferring to you.
Good copy. Thank you, Bridge.
Chris, why don't you tell us?
How far
You can
was
start, and I can always
Cool.
chime in after with
Cool.
things that may
40.
not have been said.
All right.
Wait, we were talking at the same time. What's the deal? Are you trying to get out of talking first?
Yep, she is. So it's your turn.
Okay, well, these manganese crusts,
Actually, not
which
right now.
you can't see real well in this image because there's a sediment cover over them, but we sort of know that they've got this crust on it. This is precipitate from the overlying seawater, and I believe it's felt that it's bacteriologically mediated, if you will. And it's a very, very slow process. So the rock doesn't, the natural rock doesn't look like this with all that texture in it. Instead, the rock just might look sort of a smoother basalt, and then over time,
You
this
wanna go in
manganese
on these
crust
two spot
develops
lobster?
on it and creates all this texture that you're seeing here. We don't know how thick these layers are, but we suspect it could be centimeters thick here because this is a very old seamount. So why does anybody care, or why would anyone care about these crusts? Because they are chock full of minerals that we need if we want to continue enjoying our cell phones and solar panels and wind turbines and electronics and all types of things. They are loaded with copper, rare earth metals, titanium, iron, cobalt, manganese, just tons of things in there that make them very valuable. The reason why deep-sea mining hasn't occurred until now is the technology hasn't been there, and it's been cost prohibitive because they're in such deep water. But now the terrestrial sources of a lot of these minerals, the prices are going up, the technology is being developed, so deep-sea mining is right around the corner. And as you can see, the only way to get those crusts up are to rip them off the surface of the rock, and of course, everything that's growing on them is going to get ripped up
Watch
too.
leads and shoreside. This
So
is
what we
Roland,
want to
video
do is try
engineer.
to find
We're about
very
to,
sensitive
make a
areas, for
test
example,
change.
this
We're
area,
going to take down all the streams.
where mining would not be a good
at which
idea
time,
because you've got a
we'll
really nice
lose all
community going on down here.
signal until we complete this modification. This is a test in order to
Thank you
maybe
very
regain
much,
full
Chris.
control.
So I think I'm
Stop,
not sure if
stop.
you just heard Roland.
Let Kelly do this.
Okay.
Hey, Roland, I'll go ahead and give that announcement. I don't think they heard you. You and Chris were talking at the same time. So I think everyone on shore is aware that we're having significant problems with our video feeds today in a way that hasn't happened before. And we think that's due to some modifications that were made last night that they would like to reset. The impact is that this is going to take the live video feeds down for about 2 minutes.
Yeah,
if there's a problem,
not a
maybe
lot
about
of tether
5
left.
minutes.
Here at the
So we
end.
wanted to touch
All right.
base with all the scientists
Look
participating
around.
right now to see if this is an appropriate time in the dive that we can do this.
So we must. you
Well,
must have
probably
just
better now
paused
than
after
never.
that last
I
one,
mean,
and
later.
now You're about to start the next one.
Yeah.
Because I stopped to pirouette around that.
Okay, then we're going to go ahead and take
think
these
we're
down.
moving
Diva
toward
and
you.
Deb will still be continuing to leave to lead the dive here just for the next few minutes, and we hope we will be back online and talking to you guys very shortly.
So we're offline now?
I guess so until they redial.
Yeah. So
you can look at anything you want, Carl. You're free. All right, yeah, we're moving. I'm moving toward you. You got tether.
This is really nice. The only challenge today is nowhere to really sit down.
Yeah. Much current?
No. It's very peaceful. I think I'm not sure if it's this one, but one of these spiral corals is called Magnus.
Really?
Spirogorgia.
Maybe you should confirm that.
Yeah.
So even though our feeds are going down, Deb and I still get to talk.
Which we're really good at.
Which
so it's okay.
pilots, you are going to be the only people privy to this.
We have a question for you.
Yes.
Oh, okay.
These spiral corals,
yes.
I think the one with the bigger loops, I'm not sure if it's the one on screen, is it Magnus Spirogorgia? Or?
No, so Chris actually knew these, so he's totally one up on you guys in terms of biology points.
Well, I'm sure he is.
Chrysogorgia?
So the Chrysogorgia is the family.
Okay.
So okay, you'll get 2 points for that one.
Okay.
And the genus is Iridogorgia.
Iridogorgia, of course.
Yeah. There is one with the name Magnus in it, right?
I mean, maybe.
There definitely is.
Yeah.
I like the way they're all like, oh yeah,
So
Iridogorgia.
Yeah.
Yeah, I knew that.
So if Carl finds a new species of coral,
Fish. Yeah.
Will it be Carlagorgia?
Can we call it Coral McCletchy?
Is that a Walking Dead tribute?
Yes, we can, but Carl then cannot be an author on the paper because you cannot name a new species
okay.
after yourself.
That's very interesting. I actually didn't know that.
You can be the author, and that's fine.
Is that a shrimp?
Yeah.
That is a shrimp.
Should we look at the shrimp?
Yes, let's look at the shrimp.
Okay, video, if you'd like to go in.
So this looks like one of these Caridian shrimp, which we've seen,
Okay,
I think,
hold
one of
there.
on every single dive nearly, apart from maybe one or two. Whoa.
Go in a little tighter.
Smaller.
And you can see they've got these really interesting modified pleopods, which they used to swim through the water in a really graceful manner, as well as these very, very long antennae, which are obviously used for sensory.
I think it's a good idea to put keep the ship moving another 40 meters at 240.
I think
reasons. What are you
I
giving
think we
me?
have some time. We don't want to.
Yeah, how many meters is it? What, like 100, 120?
Killing us. It's fine. It's more that I really have a hard time typing underneath.
Okay, whenever you're done.
Yeah,
Wow, I'm like
about
blind.
160.
Oh, 160. And then,
okay.
Well, how much time do we have left on bottom?
Yeah.
Let's see. So,
is that a good way to end it?
Am I on? I am on.
Mm-hmm. Okay.
Yep.
Yeah, I just don't want to stack up 3 40-meter
Right.
moves and then be asked for a sample or something.
What about some of these other ones we're looking at right now?
A little over an hour and a half.
There's a lot of coral out there.
Okay, so yeah, so we're seeing a lot of the same corals we've been
speed
seeing on this dive so far.
Needed to get
A lot
to
of
the top of the
isidids,
feature.
likely acinellas, a lot of chrysogorgids, iridogorgids.
Can you put in for polar
Yep,
basalts as well?
okay.
0.05 knots if we do a continuous move.
Right. So we're not in too much of a rush.
All right.
So I am at some sort of local high right now, and there's a fish, but we will have to figure out which way to go after this.
So we're at a depth of about 1,960, and we came across a really nice area of fairly high density corals.
What's pillow? Pill?
Yeah, PIL. I already got it in there, but thank you.
Have we seen this fish yet today?
So we can still do that. This looks like a synaphobranchid, so possibly, but we can do a snap zoom on it.
Okay, I'll sneak
I think
a
we've
little
seen
closer.
4 or 5 of these for this dive so far. And
At
I think
least
they
2.
thought it was
Okay, video. Want to do a snaps in here?
Synaphobranchus brevidorsalis. Did I make that up? Maybe.
So Scott France on shore was saying that there were a lot of bamboo corals in this area.
Yes, so those are the main bushy ones that we've been seeing.
Okay, you happy with that watch lead?
Yes, thank you.
All right, you can go wide video.
Can you hear video?
No.
I can't hear you video.
So they were saying Synaphobranchus brevidorsalis.
Make sure you're...
And I'm really I don't want to say somewhat convinced.
There you
I'm
go.
beginning to believe that some of these definitely are pillow
Have you said
basalts,
anything
not
important
all,
in the last 5 minutes?
but
No, just
several of
there's a
them
fish
do
and
appear to
that's a
have
shrimp,
the
that's
correct
about it.
morphology,
All right.
even when accounting for sediment and manganese crust.
She thought we were just ignoring her.
Why aren't you listening to me?
You got a coral or sponge or something off to your right.
One of the discussions we had
Tall one.
earlier in the
Yeah.
day was how old this seamount is. So
There
we
you go.
believe it's from the Cretaceous, and that's a
Just didn't
fairly
want you
big
to,
period of
so
time. It
yeah, there's no
basically
significant
is
downhill
somewhere
here.
between
It's just,
65 million and 145
slight
million years
flat.
old.
Yeah.
So the rocks themselves, probably pretty old. I don't think any of the corals are probably
So you wanna
65 million years old, though.
Increase the range of my
I
sonar?
definitely don't think so. I mean, maybe some of the
You got
fossil
enough?
ones which we're
Yeah,
seeing
I
on
can
the
do it.
seafloor covered in manganese crust, but I still don't think they're
That's
that
50
old.
No,
They
I would
Thank
agree.
you.
They would be
So-
really thick crusted then.
Yeah.
So your heading is like
I absolutely
225.
Yeah. That
But
looks
it's been very
about right.
interesting taking a look at
Hold on, let me
these,
steady up
at this
here.
substrate, looking at these manganese crusts. And as
235.
Chris
That
was
looks
just
pretty
talking
good, huh?
about,
Yeah,
this is
235.
sort of a
All right.
far edge
Are
of
we
this prime
about done with
crust
that
zone.
move?
And this area
Yeah,
has a
we
lot
finished
of
maybe a couple minutes
nodules
ago.
and a lot of
Okay.
ferromanganese
Yeah, are you
crusts.
happy?
And it could be an
235.
area that
40 meters at
is of
235?
interest for mining
Sure.
in the future
I'm
because
putting it
of
in.
its
Cool.
density and amount of
Bridge,
rare
the
earth
snap.
metals, as well as some more common metals that are becoming
Hi,
scarce
can I
on
get a
land.
ship move of 40 meters at
So yeah, so just to finish
235
what Chris was
degrees
saying,
at 0.2 knots?
this area is on the western edge of the prime crust
Good
zone.
copy. Thank you, Bridge.
And so this is an area where the crust is the thickest and also the most commercially attractive in terms of metal content. And
Let me know if
luckily
I can
here we're...
Could we look at?
Come down a bit.
He's back.
Yeah, we
Sorry, I
didn't...
wonder if we could look at that thing that just went off to the left there that was on the side of the rock. I couldn't tell whether that was a sponge or a very different coral. It was
Sure.
just slightly to the left of where we are now.
We thought we'd lost you, Chris, but okay. So pilots,
Yes.
you go for it.
Can you hear it just a little bit to the left? Evidently, we're looking for either a sponge or a coral that we don't know what it is.
Okay. First, I just wanted to get a quick zoom on this object in the middle. Video, could you zoom on this rock, I
Okay,
think?
zooming in. Might be a fallen
Is it another dead
sponge.
sponge?
Oh, it's a leaf.
No, so it's
Oh,
a
no.
leaf? No, we
No.
don't get leaves down here, people.
No, but they...
Oh wait,
a
it's
sunken one.
rock or something.
No.
So
What is it?
this is a sponge skeleton.
Okay, gotcha. All right, you can go wide.
Okay.
And that was
Going
not
wide.
true. We do get leaves down here sometimes.
Yeah, once in a while you see.
Yeah. Some pieces of
random.
wood and some leaves and some seagrass and other things.
Okay, turning back for a sponge on a stalk, maybe. Or was it this round one on
Oh,
the
so I
ground?
think he must mean the one in the distance, the round one quite far away.
Okay, quick zoom on this jelly video.
Okay. Zooming
Is
in.
that what that was? I was wondering what the little white thing was.
Doesn't get much more centered.
Oh, is that
So
one of
this
those
is a
dandelion
so
things?
this is a dandelion or a
Wow.
it's a type of siphonophore. Wow, look
that
at all
looks
those
really,
little
really different than